Compared to our closest living genetic relatives, humans are, physically at least, rather unimpressive. We lack strength, we can't run as fast, and we are much worse at climbing. Yet we have architecture, religion, science, language, literature, art, culture, and vision. Achievements all made possible by our larger brain. And since the writings of Charles Darwin, we have believed this development to have been constant and linear. I always assumed that the advent of modern man was inevitable, that the development of Homo habilis and Homo erectus were destined to lead us to this point. But is this really true? New findings from Dresden suggest a quite different conclusion, that the evolution of the human brain has been far less of a slow and gradual development, and more the result of a banal coincidence, which can be traced back to the sudden change of a single gene. If it turns out to be true that this point mutation is so causally significant, then you have to ask whether humans really were always destined to exist in their current form. If we summarize our various experiments and findings, it seems that we have made an important contribution to understanding how humans came to have such large brains. Homo sapiens shares almost 99% of his genetic material with other hominids. Apart from a few genetic details, what sets us apart is the size of our brains. If you think about human achievements, not only individually but also as part of a team, the moon landing 50 years ago is still something that blows my mind. Other hominids couldn't have done that. Professor Dr. Wieland Huttner is head of a research group at the Max Planck Institute for Molecular Cell Biology and Genetics in Dresden. In 2001, he was appointed director. Even as a child, Wieland Huttner wanted to crack the many unsolved mysteries of the human brain. When I was 17, I got into an argument with a boy in my class about whether we would really ever understand the human brain. He said it would always be a mystery. I disagreed. So I started thinking how I could best get involved with this area of research. I'd planned to study psychology or chemistry, thinking that either one of them would help me reach my goal. And then my father said, "Study medicine, that's right in between." So I studied medicine, but straight after the basic medical examination, I began to focus on the brain. Since then, Wieland Huttner has been using his knowledge of medicine to answer precisely these questions. How did evolution cause the human brain to grow? What biological cell processes caused this to happen? Our brains are 3 times larger than those of other ape species. Which changes in our genome led to this growth?
Es gibt sicherlich Wissenschaftler, die aufgeben.
There are scientists who give up at a certain point. I don't believe that I am one of them. In the past 25 years, we have made a lot of progress and have managed to build on this progress as well. But that's only been possible due to the fantastic colleagues I have working alongside me. 13 scientists and assistants from around the world are working in Huttner's research group and partner laboratories.
We are
what you would call cell biologists. And so, as cell biologists, we began to address the question of how our brains grow.
und haben zuerst
First, we studied what are known as the progenitor cells, which need to divide in order to produce nerve cells.
zellbiologisch untersucht.
And in the process, we noticed that they have an extremely interesting morphology.
Morphologie haben und haben dann
So we said,
gesagt, das nutzen
let's
wir
use
aus,
this in order to isolate the
diese
cells.
Zellen zu isolieren.
The progenitor cells are the first step in the development of the embryonic brain. Managing to isolate these has formed the basis for studying the next stages in the growth process. For me, the most impressive example of human brain capacity is Einstein's theory of relativity. It's incredible how concepts as intangible as time, space, and gravity can be combined in a single formula, and Einstein has thus far been proven right. Neurobiologist Dr. Michael Haider is a scientific assistant in Wieland Huttner's research group. For years, he has been involved in the search for the genetic triggers of human brain growth. The first question we asked ourselves was, which genes are actually active in these stem cells or progenitor cells of the brain? So we came up with a list of around 3,000 genes. And what we found interesting, of course, is when they first appeared, or in what other mammals they can be found. We then managed to narrow it down to 50 genes that can only be found in primates. And of these 50, 15 were specific to humans and not found in any other mammal. And one of these 15 was RGAP11B. RGAP11B. This gene, known as 11B for short, attracted the scientists' attention. It only occurs in humans. Does this gene cause the progenitor cells to multiply? These key cells are what cause the brain to grow in the embryonic phase. In 2015, the researchers began studying RGAP11B in more detail. They planted the human-specific gene in the brain of a mouse embryo.
In
the original tests, we introduced the 11B gene into mouse embryos and encouraged its expression, as we say, for a transient period, just a few days. The mice's brains suddenly contained more progenitor cells. This is the prerequisite for more brain growth. And incredibly, in half the cases, the mice's brains were not only larger, they also showed extensive folds, which is otherwise never seen in rodents. The findings from Dresden were a game-changer. RGAP11B triggered the development of significantly more basal brain stem cells in the mice, and even caused folds in the otherwise smooth cerebral cortex to occur, in line with the pronounced folds we see in humans. The researchers decided to go one step further. There are other kinds of animals that are much better suited to brain analysis, such as the ferret. So we planted the 11B gene in this species' brains as well. And the interesting thing is that, while in the mice 11B caused certain stem cells to proliferate in the brain, these were not exactly the right stem cells. In the ferret, however, exactly the right stem cells proliferated, to 6 times their usual number. Another sensational finding. Even in ferrets, which already have a folded brain, gene 11B triggered a clear increase in brain size. The folds became stronger, and the cerebral cortex thicker. Could RGAP11B be the missing piece of the puzzle long sought by scientists? The key component in the growth of the human brain, and what led us to become the planet's dominant species. For me, our greatest achievement is mastering fire. Dr. Franka Parianen is a neuroscientist, author, and keen science slammer. She believes it better to have wrinkles in your brain than in your clothes. She knows a lot about both. She also knows about evolutionary brain growth.
Man kann schon sagen, dass das, was gewachsen ist in unserer Geschichte.
What has grown most clearly over the course of human evolution is the neocortex. Our entire brain has developed, but it's the neocortex in particular that has grown disproportionately. And it's this part of the brain that enables us to do many of the things that we associate with intelligence, like understanding cause and effect, predicting the future, and so on. When I'm solving maths problems, ideally something in the front part of my brain should be active. Complex thought processes, speaking, dreaming. All of these things happen in the neocortex, the youngest part of the brain. The more furrows it has, the stronger the folding, and the more room there is for the grey cells to grow. But size does not equal intelligence. If we looked at two different people and said, this person has a larger brain and that person has a smaller brain, we'd be talking about the weight. We can't deduce intelligence from weight. But if we looked at the interconnectedness within the brain, we'd be much better positioned to draw conclusions about intelligence. In other words, we can't just think in terms of building blocks. We have to ask, how does it all fit together? How does it work together? To what extent do these connections stretch over the entire cortex? And how quickly do the connections form? How quickly can one cell react to another? Another important point is memory. How large the memory is plays a very important role when discussing human intelligence. The researchers at the Max Planck Institute in Dresden have found a human gene that causes nerve cells to multiply and the cerebral cortex to grow, at least in mice and ferrets. But does it have the same effect in more highly developed life forms? We found that RGAP11B multiplied the progenitor cells of the mice's brains and also caused the brains to grow, sometimes even with pronounced folds. We have to acknowledge that mice and humans are quite far apart, especially in terms of brain structure. That's why we decided that if RGAP11B really is the crucial gene in terms of the evolution and development of the human brain, we need a test subject that is closer to humans. Michel Heider was therefore assigned with introducing the human gene RGAP11B into the embryonic brain of a primate for the first time. We decided to carry out the first experiments on common marmosets. These are relatively small monkeys that have a smooth brain in contrast to humans and chimpanzees and other higher primates. However, these animals already have all the stem cell populations that we also find in humans. That makes them the ideal candidate for these tests. Experiments on unborn animals, carried out under strict international ethical guidelines, are essential for the basic research work of the Dresden scientists.
Die Fragen, die wir untersucht haben, wie ein menschliches...
Our questions about why the human brain has become larger during the course of evolution and also during the development of the fetus, and how this is connected to what are known as the stem cells that produce these cells in the brain, can only be answered by carrying out research on living animals. That's not possible in a petri dish. And our research also has a medical aspect, especially in the fight against neurodegenerative diseases, which are currently on the increase. If you're considering cell replacement, you need to know about stem and progenitor cells, which we have now studied in detail. And these studies can't be done on mice. The mouse has hardly any of the stem and progenitor cells that shape our human brain. We have no alternative but to study the non-human primates that are our closest relatives, the ones we're allowed to experiment on. In a Japanese partner laboratory specializing in such experiments, RGAP11B is introduced into the monkey's egg cells.
Die Prozedur an sich ist, dass man Eizellen entnimmt.
The procedure involves taking the egg cells from the monkeys and then fertilizing them in vitro, in much the same way as we do with human egg cells. Then we inject them with a virus that is carrying the RGAP11B gene. The virus then infects the fertilized egg cells and integrates itself into the genetic material of the marmoset. And these egg cells are then transferred back into the animal. We stop the process on day 101 or 102 in the second trimester, meaning the animals are not born. We terminate them in the fetal stage. This was very important for us. It takes several months before the first genetically altered monkey brains arrive from Japan. Of course, we were sent pictures of the first brains after they were taken out of the monkeys. After that, it took a very long time to get all the approvals we needed to import the brains into Germany. First, we check if the specimens were kept cool during transport. If the cooling was ineffective, then the RNA molecules can break down, for example. That prevents us from being able to detect certain things. Then we measure the standard parameters, such as the size. And then the specimen goes for analysis. We usually separate the two halves of the brain because that enables us to do two different things with them. What will these experiments reveal? First, the brains of the monkey fetuses are encased in a gel, which allows the researchers to cut them into wafer-thin slices of just 90 micrometers in width. We're mainly interested in the stem cells. If I were looking at the brain as a whole, I wouldn't be able to see these stem cells. The stem cells are found inside the brain. And that was perfect slicing, textbook stuff. Michel Heider will analyze around 1,000 of these brain tissue samples over the next few months. So in the next step, we want to color these stem cells. There are different populations of stem cells, and if we looked at the section as it is, we would just see groups of cells that we can't distinguish from each other. Making stem cells visible to the human eye is a complex process, one that the scientists here have optimized for their research. We use different markers for these neurons. We can dye the neurons that are found deep inside the brain, that are somewhat older from an evolutionary perspective, as well as neurons that are found higher up and are therefore younger. What's interesting for us is not only the number of stem cells, but also the number of different neurons. The question is whether RGAP11B causes the stem cells to multiply and therefore to increase the number of neurons. The moment of truth has arrived. The result of years of research is finally here and proves in detail what the researchers had only suspected. It's a scientific breakthrough. And the folds? Yes, you can see it here. It starts at the front and then becomes more parietal. And what becomes clearer here are the folds, not so thick at first, but then getting thicker. And here I started to measure the GI. And usually at this stage of growth, there wouldn't be any folds. It would be completely smooth. Great. And that's just in one or in several of them? That's just from one as an example, but you can see the same pattern in all the others. In all of them. To different degrees, of course, but it's always visible. The results from the marmoset brains are impressive because they show these folds, even though the brains are normally completely smooth. And we can also see that the brains are much larger than they would usually be. We can see that the relevant stem cell populations have grown. So we can observe all the things we were hoping to see. The RGAP11B gene has reproduced exactly the same nerve cells that are also present in large numbers in humans. 11B has therefore caused the brains to grow in mouse, ferret, and marmoset embryos.
Wenn man sich diese Bilder anschaut, also
When you look at these pictures, they almost look like works of art. The various colors merge into the different layers, making for very impressive images. Planning, taking action, understanding numbers, awareness of our environment and fellow humans, self-awareness. Is our large brain the sole reason for these exclusively human abilities? Where exactly is the dividing line between man and ape? Since 2001, Leipzig Zoo has been home to the Wolfgang Köhler Primate Research Center, also known as Pongoland. 4 species of great ape live here. The center provides the scientists of the Max Planck Institute for Evolutionary Anthropology closer insights into the behavior and cognitive abilities of hominids.
Ich denke, dass eine der bedeutendsten Errungenschaften
I think that one of the most important achievements of humans is education, meaning that we're capable of passing knowledge on to others, and not just from one person to another, like from a parent to a child, but also by passing knowledge down from one generation to the next in a collective and systematic manner. Dr. Katja Liebal is a professor of comparative developmental psychology. For years, she has been studying differences in the ways humans and apes communicate.
Was wir gerade gehört haben, ist ein
What we've just heard is what's known as a long call. It's a territorial call, which the primates also use in the wild, to indicate that the dominant male is claiming a piece of land as his own. And he sometimes does this to zoo visitors whom he knows well or is willing to tolerate. He sometimes throws a few things around, fortunately not in any particular direction. At that point, it's important to take a step back and not provoke him any further. Katja Liebal carried out a large part of her PhD research in Pongoland, also studying orangutans. One of them was Bimbo, who can clearly still remember her.
Ich habe tatsächlich mal eine Studie gemacht, wo ich untersuchen wollte, ob sie
I actually performed an experiment to find out whether great apes come to each other's aid, especially when they see something happening to a fellow animal, such as being bothered by a human. And the question was, how do you annoy apes without crossing ethical boundaries and irritating them too much? So what we did is offer them food, and then pulled it away and ate it ourselves. And that was enough to ensure that even after 10 years, he still can't stand the sight of me. Bimbo's anger is clear to us from his gestures and facial expressions. But not everything can be translated directly. What we know is that the eyes, for example, are not as strong a communicative signal as they are in humans. That's because the white sclera is missing. They don't seem to use their eyes as much to determine where someone is looking. You can often see that in interactions, the orangutans themselves avoid direct eye contact. In gorillas, that's also a strong signal. It can even be understood as a threat when a person stares at a gorilla. Unlike monkeys, humans communicate a lot with their eyes. The dark circle of the pupil against the white background enabled early humans to convey information wordlessly, presumably long before spoken language developed. Then came hand gestures and facial expressions, important tools for communicating within a group. Monkeys don't use abstract gestures like, it's okay, or I'm going for a cigarette. Nor do they make symbolic gestures that imitate movements or shapes. Typical chimpanzee gestures would include stretching out the arm to offer food. They also do it sometimes to beg when another animal has food. It is still often unclear what is meant by the gestures and the facial expressions of the great apes. Our tendency to project human behavior onto the ape's body language also makes objective research difficult. One difference, however, is clear.
was wir bei
One gesture we don't see the apes making is pointing with their index finger. Every gesture they make means the same thing. I want something now, meaning I want food, I want to play, I want you to leave me alone, I want to mate. But they don't use gestures to say, for example, look, did you see that bird up there? It's raining. Or, do you want to go outside? And apes are considerably worse than humans in understanding the cooperative potential of gestures. It's not in their nature to want to share information.
Es liegt einfach nicht so in ihrer Natur, dieses Teilen von Informationen.
Sharing one's own world with others. No species can do that better than humans. This new aspect to interhuman communication was an important step in our development. What makes us human is, largely, our ability to cooperate with others. When we look at large primates, it's worth asking ourselves, how do they differ from small children? We tend to think humans have a wonderful grasp of cause and effect relationships or can make complex predictions about the future, when in fact apes and humans are pretty much alike. What humans excel at is any task that requires more than one person. In other words, we're better at coordinating our work, better at working together, and that is a tremendous advantage. Human children are great at not only dividing the work, but they also understand that afterwards they have to divide the profits. So humans are good at anything that requires more than two people. And only because of this was modern man able to build what we commonly know as human civilization. If we had to manage everything on our own as individuals, we wouldn't have any time for art or for building cities or palaces or whatever. Instead, we'd be worrying all the time about where our next meal is coming from or who's going to look after our kids. And it's only because we can delegate some of our responsibilities to others that we humans are able to achieve so much. But did this only become possible after a gene we call RGAP11B triggered the significant growth of our brains? To answer this question, the researchers at the Max Planck Institute in Dresden had to study the workings of the gene in more detail. The question was no longer if, but rather how 11B created more brain cells. What the scientists already knew was that RGAP11B is the partial duplication of a gene that also occurs in other mammals, RGAP11A. This duplication led to the creation of RGAP11B. This process only occurs in humans, however, and not in any other mammal.
Die partielle Genduplikation als solche.
The partial duplication of the gene took place around 5 million years ago. But the brains of our hominid ancestors 5 million years ago were also very small in comparison. This means that this gene duplication cannot explain why our brains are so large, because they would have grown much earlier on, which they didn't. In other words, the duplication of gene 11A to gene 11B had no initial effect. When 11B came into existence some 5 million years ago, modern man was still a long way off. What humans are particularly good at is putting themselves in other people's position. We call it the theory of the mind. Other animals can do it too, but we do it particularly well. Scientists at the Max Planck Institute for Evolutionary Anthropology in Leipzig are studying the development of hominids. Dr. Philipp Gunz is a paleoanthropologist and an expert on what our earliest ancestors looked like 5 million years ago. The ancestors of the Australopithecines lived in Africa 5 million years ago. Australopithecus is a genus of hominin best known from the famous Lucy, who was discovered in the 1970s. From Lucy and her contemporaries, we know that they walked upright and lived on the savanna of eastern Africa. Not until the start of the human lineage, around 2 million years ago, do we see brain volume beginning to increase. But if, for millions of years, gene 11B failed to trigger any increase in human brain size, why did the brain's stem cells suddenly begin to multiply so quickly? The Dresden scientists found the answer to this question inside the gene itself. A minuscule change, known as a point mutation, which gave 11B a completely new function. We managed to rebuild and test the gene as it originally came to exist 5 million years ago through duplication. It doesn't do anything on its own. It is only via the point mutation that the unique characteristic of this gene, its ability to enlarge the brain, comes about during the development of the fetus. And we are convinced that this point mutation occurred much later than 5 million years ago. Probably more around 1.5 million years ago, when there was this enormous boost in brain size development in the human lineage. This sudden genetic mutation in RGAP11B was as unlikely as a lottery ticket with 6 matching numbers. Point mutations alter only one of several thousand nucleotide bases inside a gene. In 11B, this coincidence led to a change in the transcription process of the RNA, and thus to a completely new effect. Due to the mutation, 11B received the signal to migrate from the cell nucleus into the mitochondria, where the cell produces its energy. And once there, it ensured that the stem cells would divide much more frequently. Which meant, in short, that humans gained a much larger hard drive. The Dresden scientists had found the proverbial needle in a haystack, because mutations like this have taken place millions of times throughout human evolution, but mostly without any consequences. There are many kinds of mutations in the human body. Everybody carries mutations inside them, which can give rise to benign or malignant tumors. However, most mutations don't make any difference. Only very few of them actually have a positive effect, and natural selection only heeds positive changes. These are the changes that can survive the entire evolutionary process. We know that there are a lot of genetic similarities between modern humans and our closest living relatives, the bonobos and the chimpanzees. And what that means is that often very small differences can have a substantial effect. A growing brain needs more space. So how did our ancestors' bony skulls adapt to the larger brain? Here we can reach a pretty clear conclusion. This is a 3D model of a human baby skull. The fontanelle is still open directly after birth, and the bones are not yet fused together. As the brain expands, it pushes the skull outwards. At this early stage of development, the increasing volume of the brain is what drives the growth of the skull. In 2011, researchers from the Max Planck Institute for Evolutionary Anthropology managed to decode the Neanderthal genome for the first time. As the data shows, Neanderthals also possessed the mutated RGAP11B gene and had a brain of similar size to modern humans. This suggests that the point mutation of 11B must have taken place before the development of the Neanderthals.
Wir wissen, dass heute lebende Menschen und Neandertaler
We know for a fact that modern man and Neanderthals share a common ancestor, who was alive at least 600,000 years ago.
älter als 600.000 Jahre mindestens.
This ancestor was Homo erectus. The point mutation of gene 11B presumably started in a member of this species.
Homo erectus ist die erste
Homo erectus is the first human ancestor to have lived throughout the Old World, meaning Africa, Europe, and Asia.
Bis Amerika sind die unseres
They didn't make it to the Americas, as far as we know.
Begabter Jäger und
They had brains that were more than double the size of those of the great apes alive today.
also mehr als doppelt so groß als bei heute lebenden
Homo erectus was a talented hunter-gatherer and probably hunted large prey using spears.
und Jagd auf Großwild gemacht.
However, it does not appear that the results of this genetic mutation spread like wildfire throughout the species.
Ohne jetzt die Funktionsweise dieses Gens zu kennen.
Without knowing exactly how this gene works, we can assume that its effects were not particularly dramatic at the start,
Weil gegen extrem dramatische Effekte wird eigentlich
because natural selection normally filters out any extreme changes.
doppeln. Ne? Weil so ein Gehirn,
Brain volume in humans can't simply double overnight.
der also
Brains that size require a lot of energy,
abgerechnet
and energy is the currency, so to speak, that nature deals in.
Organismus so ein großes
An organism has to be able to afford such a large brain.
Glauben, dass sich Homo
We think Homo erectus could sustain a brain of this size because it consumed a very high energy diet through its highly effective hunting methods and therefore had a surplus of energy.
haben und daher eine Energie
So we can assume that the increase in brain size was a gradual change that took place in early childhood.
der frühkindlichen Entwicklung ist.
Our forefathers' direct descendants therefore had more neurons at their disposal, allowing them to develop their cognitive abilities.
Es gibt verschiedene Grundbedingungen.
There are some basic conditions that we need to meet in order to get the most out of this large brain. The first is that you have to be able to sustain the brain. That means we need the food, we need the energy, so that this brain can keep working. Then we need to network with others. That means that we really have to use the first years of our life to learn from other people, because there's an incredible amount in human society that we have to understand in terms of what we can or cannot do. Otherwise, we risk being excluded from society later on. As individuals, we will not get far on our own. The random mutation of RGAP11B was clearly such an enormous evolutionary advantage that the mutated gene was passed on to all of Homo erectus's descendants, and therefore to the entire human race. If the carrier of the mutated gene had died in the plains of Africa without any offspring, modern man would probably not exist. What the Homo genus did with this random coincidence, and what sets it apart from its forefathers, is also being studied by the primatologists at the Max Planck Institute in Leipzig.
Das Alleinstellungsmerkmal des Menschen ist wirklich die
What sets humans apart is language, symbolic communication.
des Lebewesen. Kann
No other creatures can express their feelings, their thoughts, and their wishes in the same way that we can.
sich so äußern, wie wir das jetzt können.
Dr. Horman Wittig is the director of the Chimpanzee Project in the Thai National Park in the Ivory Coast. Observing chimpanzees in their natural habitat has given researchers some insights into the evolutionary basis of human behavior.
Wir wissen, dass ein Teil unserer Vorfahren vor 7 bis 8 Millionen
Around 7 to 8 million years ago, some of our ancestors went out into the savannah.
Aufgaben stellen. Und deswegen
They had to face new challenges, and the pressures of natural selection were so strong that a substantial change took place, which ultimately led to modern man,
letztendlich
while the remainder of our ancestors stayed where they had always been, in the forest, in the tropical forests of Africa.
ist, wo sie immer waren, im Wald, in den tropischen Wäldern Afrikas. Die Selektion
The processes of selection that affected these apes were much more conservative, and that is why evolution was more limited in their case.
Evolution konservativer. Deswegen
We can therefore assume that the behavior of our ancestors some 7 to 8 million years ago was far closer to that of chimpanzees than it would be to our behavior today.
Jahren gezeigt hat,
That's why it makes sense to study these animals closely, in order to understand where we as a species come from.
damit wir verstehen, wo wir herkommen.
These studies have shown that we have much more in common with chimpanzees than we realize. They too form social units, share food, maintain friendships, show empathy, use tools, communicate, and probably dream. Groups of chimpanzees have even been shown to form their own individual cultures.
Man hat zum ersten Mal Schimpansenkulturen untersucht, indem man
We have studied chimpanzee cultures for the first time by looking at behavioral variations.
zum Beispiel, dass obwohl es
We found, for example, that although there are cooler nuts all over West Africa, only part of the chimpanzee population cracks these nuts in this area.
in diesem Gebiet diese Nüsse auch knacken. Und warum ist
And why is this?
es gibt keine ökologische
There is no ecological reason for it,
es gibt keine
and there's no genetic reason for it. So what is the reason?
Hier zeigen die
It's how the chimpanzees show which social group they belong to, which culture they belong to,
welcher Kultur sie gehören.
just like we do when we eat rice with chopsticks, or a fork, or with our hands.
Oder einer Gabel oder mit den Händen essen.
Wir gehören ja auch zu den wenigen.
We're the only species that carries tools around all day, and not just our mobile phones. In the past, it was a pocket knife, a watch, even our clothes or glasses.
Wir tragen Sachen,
These things become a part of who we are, so our culture is part of who we are.
uns selbst.
But while the cultural evolution of our ape cousins came to a standstill, that of humans continued to develop exponentially.
Also wenn wir uns unsere
If we looked at our evolutionary development as a curve, it would go like this.
Und das ist,
And that's something that Darwin alone cannot explain. This idea that the current generation must die out before we can take a few steps forward, and then after that the next generation must die out, that won't help us to develop at the same rate we have managed to over the last 40,000 years. And all those things that we associate with humans, religion, culture, tools with small moving parts, these are all things that have been created in this very short period of time. The reason that we've evolved so fast as a species is that we don't have to invent everything from scratch each time. An engineering student doesn't have to figure out on their own how a steam engine works. We can build on knowledge that has been gained by others. So, in a sense, cultural evolution is our evolution.
Kulturelle Evolution, unsere Evolution.
From I to we. Only humans are capable of uniting hundreds of thousands of their own species under a single idea. Only humans have managed to bring together and organize millions of others in one place. Our social intelligence was one of the key factors behind our remarkable development as a species.
Die Idee ist, dass die soziale Intelligenz
So we believe that our social intelligence is not just a byproduct of our evolution, of our growth as a species, but actually one of the driving forces behind it.
dahinter war. Und das Ganze wird dann
And the whole thing is then taken a step further by the cultural intelligence hypothesis, which basically says that the passing on of this cultural information, such as the mastery of fire and so on, is what has caused our brains to grow so much.
Informationen möglich gemacht wurde. Was sind denn unsere
So what are our evolutionary advantages? We don't even have fur. We need to wear clothes to survive. We don't have claws or especially strong muscles. In fact, we have lost much of our original muscle mass, so we have the energy to feed our incredibly powerful brains. We're not built to survive on our own, but instead we've created a community that we can rely on.
Wir denken uns immer,
We often think we've had enough of other people and that it would be nice to withdraw to a desert island somewhere. But we wouldn't survive on a desert island. We wouldn't manage to meet our own needs and we'd get very hungry. And that's the point. The community is a part of who we are, and we would not exist without the community.
die Gemeinschaft.
Living in groups provides protection, sustenance, and a sense of belonging. But it also creates competition for food, territory, and reproductive partners. This can cause friction and aggression.
Diese Konkurrenz muss irgendwie geregelt werden.
This competition needs to be managed in some way. Not everyone can eat the same piece of fruit. We have observed how chimpanzees follow a kind of decision-making process. They ask themselves, should I accept that another animal has this piece of fruit and that I can't have it? Should I share the fruit? Or should I hit him and steal the fruit for myself? Our early ancestors had different ways of resolving aggressive conflicts. Similar methods can be observed in chimpanzees to this day.
When
chimpanzees get into conflicts over borders, a number of different things can happen. The first thing is that they detect the other animals and a loud fight ensues. They beat the roots of the trees and make pant hoots and pant roars. These are very loud calls that can be heard for miles around.
Über Kilometer hinweg zu hören sind.
And usually these battle cries are enough to determine which of the two groups should retreat.
Wer von den beiden Gruppen den Rückzug antreten muss.
But sometimes this is not enough, and the chimpanzees confront each other to show how big their group is, who is taller, who is stronger, and so on.
Wie groß man ist, wie stark man ist. Und dann kann es eben zu diesen direkten...
And sometimes these direct physical fights take place, which can result in injuries or even deaths.
Oder in Tötungen ausarten können. Was man bislang allerdings noch nicht beobachtet hat.
What we haven't observed so far in these fights, however, is the use of weapons.
Auseinandersetzungen Waffen verwendet worden sind.
Although chimpanzees understand how to crack a nut using a stone, we've never seen them use weapons like this in their conflicts with other animals.
Diese Waffen nie beobachtet worden in einer Auseinandersetzung mit anderen.
Unlike humans, conflicts are a part of everyday life for all primate groups. Waging war with deadly weapons, however, is an invention of modern man. This is the other side of our species' social and cultural cognition. Can the genetic researchers from Dresden imagine apes with the ARGAP11B gene also becoming like humans in this respect? Under Research Director Dr. Wieland Huttner, this question will never be put to the test.
Nein, Versuche an den Menschen.
No, tests on apes are not allowed, and with good reason. These animals are very close to us, and I think it's absolutely right that they should be off limits for such experiments. I think it would be highly irresponsible to manipulate orangutans or chimpanzees or gorillas in this way. How old is Tony now? Tony's 28. He was born on the 13th of December 1991. Dr. Huttner also rules out any further application of their research results, either for apes or for humans.
Was für mich als Mediziner absolut aus.
What is out of the question for me as a medical scientist, I repeat, absolutely out of the question, is any attempt to, quote-unquote, enhance human brains by, let's say, smuggling in a second 11B gene or a more effective 11B gene. I would consider this to be highly irresponsible. You have to be quite clear. Bigger brains are not necessarily better. My research has been about finding out why we have a large brain. I think this is a fundamental question because it helps us to understand why we humans are the way we are. But to find some way of applying these findings, no. Although the findings from Dresden won't be tested on living people, the results are so compelling that they leave little room for doubt. Yet the researchers are still looking for further proof of their theory.
Wir haben den Beweis, dass 11B.
We now have proof that 11B can increase the size of the brain. That's what Michael Haider's work has shown us. But there is no data so far to tell us what happens if 11B is missing in humans. Would these people have smaller brains? There haven't been any humans in whom 11B is completely lacking. That in itself tells us something.
Das sagt ja vielleicht auch was.
Ich denke, weiter
I don't think we can go any further in terms of introducing this gene into other organisms. Another way, of course, would be to look for patients who either have mutations in RGAP11B or some kind of loss, and to see what their brains look like. This could, of course, be done non-invasively using MRI or something like that. A way of using their findings will be left to others to figure out over the coming years, or even generations. But Michel Haider's research work is far from over. We don't just have this one gene. Michel Haider has discovered 14 others that are specific to humans and that are highly active in stem cells. There's sure to be a backup. And another interesting question for me is, there are 15 genes. Are there natural interactions between these genes? Are there networks that overlap? How do these genes interact with each other? This is another exciting question that remains to be answered. If the research findings of the Dresden scientists can be substantiated over the next few years, Wieland Hüttner could one day experience the completion of his life's work. A comprehensive theory about the growth of the human brain, an essential piece of the puzzle that is human evolution. A world-changing coincidence that, many hundreds of thousands of years ago, made the miracle of Homo sapiens sapiens possible.
Speaking words of wisdom, 11B. Speaking words of wisdom, 11B.